Hello, everyone, and welcome to this week's seminar of the Living Earth Collaborative and Washington University's Evolution, Ecology, and Population Biology program. I'm Matthew Austin, a postdoc with the Living Earth Collaborative. It is my pleasure to introduce our speaker for this week, Dr. Theresa Gildner. Dr. Gildner is a human biologist whose research focuses on how lifestyle and environment affect health outcomes, ultimately with the goal of identifying health disparities so that effective intervention strategies can be designed. Dr. Gildner has an impressive record with myriad publications in high-impact journals such as PNAS and global collaborations that span the Amazon to the southern US. Speaking to the global importance of her research, Dr. Gildner has worked extensively with the World Health Organization's multi-country study on global aging and adult health. And as a postdoc, she co-founded the COVID-19 and Reproductive Effect Study, which examines the impact of COVID-19 on healthcare and birth outcomes among pregnant individuals in the United States. This effort has already resulted in multiple peer-reviewed publications. Dr. Gildner is a recent addition to Washington University, having joined the Department of Anthropology as an assistant professor just January of this past year. Prior to joining WashU, Dr. Gildner received her master's and PhD in biological anthropology from the University of Oregon, and subsequently was a McKinnon postdoctoral fellow at Dartmouth College. Today, Dr. Gildner will be speaking about her research on the impacts of parasitic infection on growth, cognitive development, and economic productivity in two communities, the Shuar, an indigenous population living in the Amazonian Ecuador, and children living in a rural community in the southern US. For this work, Dr. Gildner has visited Ecuador 5 times, living in the rainforest for months at a time. And before we begin, I would like to mention that our speaker for next week will be Dr. Carlos Cadena, the graduate student invited speaker for this semester. Finally, please leave your comments and questions for Dr. Gildner in the live chat, and we will relay them to her after the talk. And with that, welcome, Dr. Gildner. I will hand it over to you.
Wonderful. Thank you so much for the kind introduction, and thank you for the invitation to present my work. I'm really excited to interact more in the future with the Living Earth Collaborative community. And today I'll be discussing my research on the relationships between lifestyle variation, environmental interactions, and parasite infection patterns in 2 contexts. First, among the Shuar, a large indigenous population living primarily in Amazonian Ecuador. And second, I'll present some preliminary results from my more recent work looking at these patterns among children living in a low-resource rural community in the southern US. I'd like to begin, though, by framing my work. So I'm a biological anthropologist, but more specifically, I'm a human biologist. So my research explores how human evolution, behavior, and social factors interact to influence our interactions with the environment and subsequent disease risk, especially parasitic disease. And my work uses a biocultural approach to examine how variation in living conditions impacts human biology and immune function. I'm really interested in understanding how these interactions can be used to design more effective disease intervention programs. And the image here is a screenshot of one of my microscope slides, which I examined for science of parasite infection. If you look carefully, you can see a couple eggs here. I'll get into that more later. But I like this image not only because it's rather pretty, but because it represents a health outcome that's determined by an interaction of the factors I'm interested in. Parasitic disease risk is shaped by a long coevolutionary history with parasitic species, by individual biology and immune system activity and development, but also by behavioral and cultural practices that influence how we interact with the environment and are exposed to sources of infection. And this is the type of integrative perspective that I believe is needed to construct a more complete picture of human illness and determine how it's best treated and prevented. In this presentation, I'll be exploring how ecological and social factors influence parasitic disease risk in 2 populations. I'll begin with some background information on these topics, focusing on particular what's known about how individual characteristics and living conditions might influence parasitic disease risk. And I'll present the results of 2 research studies I've conducted looking at how lifestyle variation impacts parasitic disease patterns. The first is the culmination of several field seasons in Ecuador, and the second is some very preliminary data from my newest project in rural Mississippi. And both of these projects have unfortunately been disrupted by the COVID-19 pandemic. So the data I'm presenting is like a couple of years old, but I do plan to go back as soon as I'm able to do so safely. So I'll conclude with a brief discussion of future research plans once I'm able to safely return to the field. Parasitic disease, which I'm primarily concerned with, represents an immense yet underfunded global health concern. So to fully grasp the costs associated with these conditions, there are some important points to keep in mind. First is disease distribution, which you can see here on the map with the darker colors representing areas of more intense infection. And as you can see, most parasitic disease often is thought to occur in less affluent countries, especially those in tropical regions. And because of that, and the fact that these conditions often receive relatively little funding from the global medical community compared to other infectious diseases, they're often referred to as neglected tropical diseases by those of us who study parasitology. And while relatively few people die from these illnesses, these conditions can often impair physical and cognitive function and may result in suboptimal growth and development patterns, which can negatively impact day-to-day life and have implications for long-term health patterns. But thinking now about infection risk, parasitic disease risk is shaped by many factors, and this includes individual age. And research, including my own, has consistently shown that children tend to harbor the highest parasite loads. And this has been attributed to the fact that children's immune systems are still developing, but also the fact that kids tend not to have the most hygienic behaviors. So this can result in increased contact with sources of infection in the environment and also support child-to-child transmission in classroom settings or during play. Over time, these high parasite burdens in children tend to decrease, especially in areas where infection's endemic, so children are constantly being re-exposed to infection. So over time, these children tend to develop some partial immunity and also hopefully develop better hygiene practices as they get older. But the high disease burdens in this age group can have severe health effects related to growth and cognitive development and deserve the attention of the public health community. And then finally, it's important to consider the economic costs of these infections. The nations that experience the highest rates of parasitic disease often lack resources to effectively study and treat these illnesses. And so this can result in a cycle where the financial costs and long-term physical consequences of parasitic infection may contribute to existing structural health and wealth inequities. So increased research investment is really needed to address health disparities, but also to support continued livelihoods. So before moving on to how my work addresses issues related to parasitism, I want to clarify what I mean by a parasitic infection. And a parasite is any organism that relies on a host, meaning it derives some or all of its essential nutrients from the host and is therefore considered to be harmful. Evidence indicates that parasites have infected humans and human ancestors for over 500 million years. So due to this long period of coexistence and the fact that pathogens like parasites evolve relatively quickly, parasitic disease has likely played an important role in human evolutionary processes. And in fact, an entire branch of the human immune system appears to be designated to dealing with these types of extracellular infections. And there's a large number of parasite species that are known to infect humans today. And these range in size from tiny protozoa, like the malaria parasite, Plasmodium, you can see pictured here, which use human red blood cells to reproduce, to much larger parasites that live outside the human body, referred to as ectoparasites, such as ticks, lice, and fleas, but still rely on the human host for nutrients. And between these two size extremes is a whole range of other parasitic species, including, for example, Giardia, which you can see pictured here and you may be familiar with or heard of, especially if you're an avid backpacker or camper. And this is a parasite that's typically acquired by people drinking unfiltered water. There's also a special group of parasites called soil-transmitted helminths that I'll be focusing on in this talk. And soil-transmitted helminths are intestinal parasitic worms that humans acquire through fecal-oral contamination. In other words, that means that they spread through water, food, or soil that's been contaminated with fecal matter from infected humans or other animals. And these infections are extremely common globally, and the Center for Disease Control and Prevention estimates that millions of people around the world are infected with a few key species, namely large roundworm, Ascaris lumbricoides, whipworm, Trichuris trichera, and hookworm, for example, Indicator americanus. And helminth infections mostly don't have severe health effects, but heavier infections can cause negative health outcomes such as nutritional deficiencies, anemia, impaired growth, diarrhea, and if in case of extremely heavy infections, you might see things like intestinal blockage, organ failure, and sometimes death. And soil-transmitted helminth infections are very common among the populations I work with, so I'll focus primarily on these types of parasites today. So now I'd like to turn to my work, identifying key determinants of parasitic disease risk. Specifically, my research tests how socio-ecological factors influence parasitic disease risk among populations characterized either currently or historically by both heavy infectious disease burdens and also by variation in living conditions. And in particular, and likely of interest to this audience, I'm interested in how lifestyle variation influences environmental interactions and associated exposure to sources of infection. This research I'll be talking about today was conducted, as I mentioned, among Shuar of Amazonian Ecuador, and also a rural community in the Mississippi Delta with the following objectives. First, to determine how lifestyle differences influence parasitic infection among the Shuar. And second, to measure current parasite infection patterns and assess links between environmental factors, parasite infection, and intestinal inflammation among children in rural Mississippi. I'll begin with my work documenting links between living conditions and parasite infection patterns among indigenous Shuar of Amazonian Ecuador. This work was conducted as part of the Shuar Health and Life History Project, of which I am part. There'll be a link to the study website on the next slide if you're interested in learning more about this project. And this specific study was published last year in PLOS One if you're interested in reading more about it. The Shuar are a large indigenous population of about 100,000 individuals residing primarily in Amazonian Ecuador. So to orient you, I've got a couple of maps here. So in the top left corner, you can see a map of South America. Ecuador is in red there. And then the middle image is the country as a whole. The area in darker green in the southeastern part of the country is the region of Morona Santiago, where most Shuar live today. And this region reaches from the eastern foothills of the Andes to the Peruvian border. And it's divided by the Cutucu mountain range, which extends from north to south. West of this mountain range is the Upano River Valley, which is an ecologically distinct area where most Shuar reside today. And then to the east, the Cutucu mountain range is the less densely populated and lower lying cross Cutucu region, which is accessible generally only by boat travel on the Morona River, as you can see in the image in the bottom right-hand corner, sometimes by journey on foot or occasionally by small plane. In the past, in both of these regions, Shuar lived in scattered household clusters and were reliant on a mix of horticulture, fishing, hunting, and foraging. And these subsistence strategies continue to be relatively important in the more remote cross-Cutucu region that's cut off by that mountain range, but are now generally less important in the Upano Valley area. And this is due in large part to changes in local level of economic development or market integration. And market integration is defined as the degree to which people consume from and produce for a global market economy. And this process is typically associated with a suite of social and cultural changes which differ by population. But common changes associated with market integration include things like an increased consumption of processed market foods, agricultural intensification, and greater travel. So these are all factors that can influence how people interface with their environment with implications for health. And among the Shuar, market integration generally differs between the Upano Valley and the cross-Cutucu regions. The Upano Valley communities are typically located geographically closer to market centers, and this allows individuals living in these areas to go into town to sell agricultural products, to access wage labor, to buy and consume market foods. So for example, the image on the left is a picture taken in the Upano Valley community of a road being constructed, so increasing that connectivity and facilitating travel into town to access these goods and services. But you can also see other signs of market integration in this image. So you can see light posts, and these electrical wires bringing electricity into the communities. We're also seeing internet being introduced into some of these communities, so a lot of rapid changes happening. In contrast, the cross-Cutucu Shuar generally still live hours away from the nearest market center, although market integration is occurring in this region too, and we are also seeing a lot of changes. But in general, many communities in the cross-Cutucu region still are many hours from a market center, and so it can be very, take a lot of time and be very costly to get into town. And so Shuar in this region tend to rely less heavily on market-produced goods and services, as you can see in the image on the right. So relying more on those subsistence strategies I mentioned earlier, so foraging, horticulture, hunting. But it's important to note that measuring market integration is really complex. So this is a multidimensional process that really varies across contexts. So it's heavily shaped by cultural, geographical, and biological factors that are unique to each population. So it's important to consider how this process is playing out within a particular setting and measure it accordingly. And market integration is also very important in the study of human biology and disease risk. And in humans, disease risk is shaped by several socio-ecological factors tied with market integration, including diet, activity patterns, living conditions, and access to services like healthcare. And many of these factors are determined in large part by the area where an individual resides. Whether that's a community with access to more market-produced goods and services, or a rural community that relies primarily on traditional subsistence strategies. And here I'm using the word traditional as the Shuar, the study population, define it. And the Shuar use the term traditional to refer to subsistence activities and lifestyle patterns that were common decades ago, prior to the 1970s, but are generally less prevalent with increasing rates of market integration. And so these so-called traditional lifestyles would include, for example, houses that are constructed with palm wood and dirt floors, and a diet reliant on a large part on fishing and hunting, foraging and horticulture, as you can see here in the image on the right. Conversely, the picture of the house on the left is a Shuar household in a more market integrated Upano Valley community. And so you can see the wood floors, the wood walls, there's a stove in the back there, and food preparation areas off of the ground. So these various factors can influence disease risk. And when studying human disease, it's important to consider the context and how market integration may be shaping interactions with the environment and exposure to disease. And for example, prior to the work I'll be presenting today, some of our earlier work in the Shuar Health and Life History Project, we did find that market integration seemed to be shaping parasite infection patterns. Specifically, cross-Kutiku Shuar were generally found to have heavier parasite loads compared to the more market integrated Upano Valley Shuar. And this might be because the cross-Kutiku communities, since they're isolated, more isolated by the mountain range, may have less reliable access to medical care and also live in less market integrated households, which may expose them to more sources of infection. But there's a great deal of variation in market integration apparent within those two regions, and even within communities. So it's really necessary to consider various aspects of market integration at the household level. So to better examine how market integration influences health outcomes among the Shuar, we measure market integration using style of life indices that were developed for use specifically among the Shuar based off previous work done in other populations and other contexts, with modifications in accordance with recent lifestyle and economic changes. So we're measuring factors that are relevant to Shuar and Shuar lifeways. And here I'm presenting the housing style of life index that we use. This is part of a household questionnaire that we go through with the participants. So as part of the survey, the participant will go through each of these list items, each measuring a different aspect of household infrastructure, and provide a number. And then at the end, we can total up all of the numbers from these line items to get a final composite score of household market integration, at least as measured by these elements of infrastructure, with a higher final score representing a higher overall level of household market integration. To measure how market integration is associated with parasite infection, I collected and processed stool samples based on previously established methods. Specifically, I handed out plastic cups inside bags. The cups were marked with participant name and identification number, and the containers were returned the following morning with a small amount of fresh stool to a centralized location where the research team was staying. When we requested that the samples be returned within an hour of passage, and I processed them right away. And this was to ensure that the samples were fresh, so I was able to visualize any parasites that were present without experiencing degradation. And I used a Kato cat's thick smear prepared from each of these specimens using a 42 milligram plastic template and stained cellophane, which allowed me to visualize the eggs better. And once dry, I used light microscopy in the field to examine the slides that same day for any observable intestinal parasite infections that can be found in stool. And I found mostly large roundworm, Ascaris lumbricoides, and whipworm, Trichuris trichuris. So I'll be focusing on those. And the images here highlight this whole process. So the middle image, you can see me processing the samples, making those Kato cat's thick smears. Image on the left, examining them. And then the image on the right is another example of what those microscope slides look like under magnification. And from the slides, I calculated Helminth egg presence, species presence, and species-specific eggs per gram, or EPG, of feces. And I want to take a moment to explain eggs per gram, because that's a really important measure for us. Eggs per gram, or EPG, is calculated by counting all of the eggs that are present in that 42 milligram sample and then multiplying by 24, which gives you a total eggs per gram. So you can see an example of this in the image on the far right, the microscope slide. So this image is just one screenshot of one small frame of the slide. And you can see several eggs here. So each of those yellowish green circles is a roundworm egg, a large roundworm. And so we go through frame by frame, counting up all of the eggs that are present for each species in the sample, and then multiply by 24. To give you an idea, the eggs per gram, the EPG values among the participants in this study who were infected, so who had at least one parasite egg in the sample, the EPG values range from 24, so just one egg in the entire sample, to 129,336. So quite a bit of variation in infection. And what this value represents is infection intensity, or the number of worms that an infected individual has. So in other words, more eggs that are present in that stool sample are indicative of more adult worms living in the gut producing those eggs, which suggests a longer lasting chronic infection. In addition, I calculated prevalence rates, or the proportion of individuals who were infected. And all these measures are of great interest to the Shuar, who are very concerned with soil transmitted helminth infection and are very aware that this is an issue in the area. So as part of our collaboration with the community, we work with local healthcare providers and pharmacies to help facilitate the treatment of individuals at no cost to participants. So returning now to my first objective of documenting parasite load as it relates to household factors, I expected that parasitic infection would be highest in less market integrated households, especially those with housing style of life factors, so specific aspects of infrastructure that have been more closely linked in the literature to parasite infection pathways. This would include having a dirt floor, not having a designated bathroom, and being reliant on unfiltered drinking water from a river or stream. For this study, I collected stool samples from 620 participants living in 152 households across 10 communities, so 5 Cross-Kutuku and 5 Upano Valley communities. And this sample allows me to get at the range of market integration that's evident within regions and communities and to better understand the nuanced associations between household market integration and parasite patterns. I should note, though, that this is unfortunately not a random sample. As with field studies working in these types of conditions, it's very difficult to draw a truly random sample. We're constrained to working in communities where we've been invited to work and that are interested in collaborating with us. And then also, once we are invited into community, we are careful not to exclude anyone who wants to participate. We open the study up to everyone. So given these considerations, it's very difficult to draw a truly random sample. But this is a large sample, which helps to capture the variation that's present in the population. And participants range in age from 6 months to 86 years old, so quite a range there too. From these samples, I calculated the eggs per gram, or EPG values of the 2 common helminths, or soil transmitted helminth species I mentioned, the large roundworm, Ascaris lumbricoides, and whipworm, Tricharis trichera. Given the nested structure of this design, I used multi-level regression analyses of individuals nested in households to test the relationship between participant style of life scores and infection status, so a binomial variable infected, yes or no. And then to examine associations between infection intensity values, those eggs per gram, and so the style of life scores, I used zero inflated negative binomial regression models given the zero inflation present in infection intensity data and in accordance with other studies. So by this, I mean the fact that we are picking up a lot of zeros in the data. So a lot of people who look like they're not infected. Some of these are true zeros, so people who are in fact not infected, but there are also methodological limitations. So we might have participants who are in the pre-patent phase of infection, so they're infected, but the worms they're infected with aren't sexually mature and producing those diagnostic eggs in the stool. Or it might just be that we're only collecting 1 sample per participant due to personnel and time constraints. And so due to uneven shedding, we might not be seeing eggs in that specific stool sample, even if the person's infected. So, and to account for this, we use these zero inflated negative binomial regression models to look at relationships between the infection intensity variation and the style of life variables. And all the models controlled for participant age, sex, and key community characteristics linked with infection risk and healthcare access, namely travel time to the nearest market center and community size. First, some descriptive numbers to give you an idea of overall parasite infection prevalence, or the proportion of individuals who had at least 1 helminth. As you can see, parasite burden in this population is quite high. So about 63% of our participants were infected with at least one of these 2 soil-transmitted helminths. And we can break it down by age too. So about 67.5% of children were infected versus 57% of adults. Children here are defined as participants under the age of 15, which is the local age of consent. And there were 329 children in the sample and 291 adults. While overall infection prevalence was quite high, these values did vary across communities, which you can see in the figure here. Communities are listed in order of median household style of life score, which this is an approximation of community market integration level, though of course, as I mentioned, there's variation within communities. This is listed from communities 1 through 10. Community 1 here has the lowest median score, while community 10 has the highest market integration, more market integrated score. So the model results indicate that participant age and travel time to the nearest market center were significant predictors of infection risk. So this indicated that younger individuals and those who lived within an hour of a market center, who lived closer to town, had higher odds of infection. The composite measure of household construction was also significantly related to infection risk, such that participants who lived in houses with higher composite measures of household construction as indicated by those aspects of household infrastructure, so had more market integrated households overall. They had lower odds of infection as expected. In addition, bathroom type and water source, so those specific pathways, the specific aspects of market integration I thought might be driving infection, bathroom type and water source were non-significant predictors, but floor material was. And this suggested that participants living in households with floors made of milled lumber had lower odds of infection compared to individuals living in homes with dirt floors, as predicted. Turning now to infection intensity or number of parasites an individual has, we can see some variation across communities here with these violin plots. So the white circle marks the community median, and the overlaid shape reflects the probability density of data at different eggs per gram values. Age was a significant predictor in these models as well, suggesting that younger individuals had higher infection intensities. Likewise, that composite measure of household infrastructure was significant, indicating that participants living in houses with greater overall level of market integration had lower Ascaris lumbricoides infection intensities, as hypothesized. Then turning to those specific pathways, the specific aspects of household construction, only water source was significant, and it signaled that participants using water from a well or piped in from a spring had lower Ascaris lumbricoides infection intensities compared to participants who were reliant on unfiltered water from a river or a stream. And again, this is as we expected. And now considering whipworm, Trichuris trichura infection intensity, participant age was still a predictor, again, showing that younger individuals had higher infection intensities. Travel time to the nearest market center was also significant here, indicating that participants who lived within an hour of the market center had higher infection intensities compared to individuals living an hour or more away. And as expected, that composite score of household construction was related to whipworm infection intensity, signaling that individuals living in houses with greater levels of market integration had lower infection intensities as expected. And finally, neither bathroom type nor water source were significant, but floor material was. And this indicated that participants living in houses with floors of milled lumber had lower whipworm infection intensities compared to those in houses with dirt floors. So putting this all together, these findings support the first hypothesis and indicate that more market integrated households, as indicated by scoring highly in that composite measure of household construction, had lower odds of infection and also lower Ascaris lumbricoides and Trichuris trichura infection intensity values. However, the study only provided mixed support for the second hypothesis. So results do suggest that water source and floor material may be especially important sources of soil transmitted helminth exposure among the Shuar. And these findings are consistent with what's known about transmission pathways. So these microscopic eggs that are present in dirt on the ground, if you have a dirt floor in your house, or in unfiltered water, may result in infection or reinfection through hand-to-mouth transmission, the contamination of food items, or drinking this unclean water. However, unexpectedly, bathroom type was not a significant predictor in any of the models. That might be because 98% of the participants in the sample used bathrooms located outside of the house. So sometimes that was a latrine, sometimes that was a community shared outhouse. But even within the more market integrated Upano Valley communities, there's not a lot of indoor plumbing in these settings yet. So I would like to address this in the future by collecting data at higher levels of market integration, Shuar living in the city where there is indoor plumbing, and perhaps then we could see some more variation in how bathroom type may be influencing these relationships. There were some interesting differences in infection patterns between the two species, between the large roundworm Ascaris lumbricoides and whipworm Trichuris trichura. So specifically, water source appeared to be a significant predictor for the Ascaris lumbricoides infection intensity, whereas floor material seemed to be more important determinant for Trichuris trichura infection intensity. And these findings might suggest that the concentration of parasite eggs in the environment may vary by species. So I need to do environmental sampling of soil and water sources in Shuar communities in the future to quantify these differences. These findings have several important implications. First, these results indicate that market integration processes are complex and they're not uniformly influencing helminth infection risks. So what I mean by that is that increasing market integration at the household level, whether that's looking at the overall composite score or these specific aspects of having a wood floor, having a cleaner water source, do seem to be perhaps protective against infection and heavy infections. But if you look at the broader community level, there were some indications that perhaps living closer to town might increase your risk of infection and also result in heavier whipworm infection intensities, which is interesting. So this seems to suggest maybe that there's these nuanced opposing effects that might be going on in terms of market integration in relation to disease risk at these different levels. So a more nuanced perspective of how market integration may be shaping disease patterns is required in future studies. And second, these findings highlight style of life factors that are more closely associated with soil-transmitted helminth infection. So the provision of an anti-helminthic treatment, which is a common public health information or intervention, is really only a short-term solution to these issues due to high rates of reinfection. And so the results presented here could suggest certain aspects of household construction that could be targeted in future interventions to decrease these relevant parasites in these communities. And if there's limited resources to allocate towards improving household infrastructure, then perhaps targeting floor material water source might be good places to start. So I now like to present some very preliminary findings from my newest project here in the United States. This project builds on my work in Ecuador and is conducted in collaboration with a Shuar Health and Life History Project collaborator, Dr. Theresa Gildner, who's a professor at University of Colorado, Colorado Springs. Specifically, we noted that many of the factors that are linked with parasite infection risk and prognosis in Ecuador also appear to be issues in low resource and underserved communities within the United States, especially underserved communities of color. Specifically, a lack of access to key health determinants, so reliable access to medical care, to functional sanitation systems, to adequate nutrition, for example, can both increase parasite exposure and also compound the negative impacts of infection in the US. But these associations haven't been well studied in the last few decades in general in the United States. So our work is testing these patterns. And the preliminary findings I'll discuss today will be submitted next month in an article that is being led by my collaborator, Dr. Theresa Gildner, to an invited special edition of the American Journal of Physical Anthropology on biocultural perspectives on disease and pandemics, past, present, and future. So I'll give you a little taste of what that paper will focus on. The idea of parasite infection, the idea that it might still be present in the United States and also in other high-income countries, is encapsulated in this concept of blue marble health. So parasitic infections have been historically widespread in the US, especially in the South, where high poverty rates and environmental factors like soil conditions and climate can favor infection spread. But most Americans today seem to believe that these diseases are now confined to lower income countries. But there's been more attention recently in the past few years on the threat that may still be posed by parasite infection in the US and other wealthy nations. And this includes work by Dr. Peter Hotez, the Dean of National School of Tropical Medicine at Baylor College of Medicine, and his concept of blue marble health, which he outlines in this book of the same name. And blue marble health recognizes that widespread poverty-related neglected diseases, including parasite infections, may occur predominantly in lower income individuals living within high income countries with major global economies, but that also have large wealth and health disparities. So this redirects the focus from that earlier slide where I was talking about how these may be confined to lower income countries, and redirects focus to these low income, low resource communities, even if they reside within wealthier countries, that this might still be an issue and we might still see high rates of infection. And this concept was originally conceived based on observations and work conducted in low income areas of Texas, documenting risk factors linked with parasite infection risk in this context. Recent work in the American South, a little bit the research I'll be presenting here, but there are also other researchers working on this in Texas and Alabama. There have been some papers published recently, especially using environmental samples that indicate signs of continued presence of soil-transmitted helminths, such as hookworm, in this region of the country. If you want to learn more, for example, you could look up some recent work by Dr. Rogelia Mejia, a doctor and professor at Baylor College of Medicine, who does a lot of work on this as well. And approximately 12 million Americans are, according to one estimate, are thought to be infected with neglected tropical diseases. And these are thought to disproportionately impact communities of color and contribute to the development of long-term health inequities due to those chronic health effects I mentioned earlier. However, soil-transmitted helminths represent just one factor that can influence individual immune function and intestinal health. And other environmental exposures, including exposure to environmental toxins and other pathogens, including harmful bacteria, should also be considered. And importantly, the cumulative long-term effects and health inequities associated with parasitic disease and exposure to other pathogens in the environment provide an example of how environments become embodied. An embodiment refers to the process by which lived experiences impact individual biology. So for example, increased exposure to pathogens, chronic psychosocial stress, unequal access to resources such as healthy foods, clean water, and effective sewage systems. All these factors are linked together, and they're also all linked with experiences of discrimination. So as outlined by the image here, you can see these complex interactions. Through these lived experiences, it's thought that inequality can literally get under the skin and impact individual biology. So in this context, impacting inflammatory immune responses and ultimately contributing to poor health outcomes. To take another relevant example that's often talked about in this types of work is environmental enteric dysfunction. And so this is characterized by elevated gut inflammation and changes to intestinal permeability. And this condition is often discussed in low resource regions and is thought to be linked with many factors that also impact parasitic infection patterns, including micronutrient deficiencies, undernutrition, limited access to filtered water and sanitation systems, and gut microbiota disruptions. You can see all that laid out here. In turn, intestinal inflammation and dysbiosis, by which I mean disturbances to the microbial communities associated with environmental enteric dysfunction, have been linked with negative health outcomes such as reduced adult stature. So cumulatively, these negative developmental effects can contribute to existing health inequities within affected populations. And this points to a greater need to focus on understanding developmental trajectories, so how these may be playing out early in life when the immune system's developing during key growth periods, and how this may then help shape later health inequities developing, with implications for lifelong health. So to explore these complex associations, the present study was conducted in a rural community located in the Mississippi Delta. This research was conducted as part of the Rural Embodiment in Child Health, or REACH study, which aims to understand how infectious disease exposure, especially to intestinal parasites, affects child health, growth, and development in low-income and rural regions in the United States. And while what I'll be presenting today is largely a descriptive set of analyses, we also hypothesized that we would observe intestinal inflammation levels in this setting that's higher than those documented in high-income reference populations. And we also expected to find signs of parasitic infection in the sample. These preliminary data were collected in July and August of 2019. The study community has a population of about 2,000 individuals, of whom approximately 95% self-identify as Black or African American. The community is also characterized by a median household income of about $20,000 and had limited access to key resources. So for instance, the nearest grocery store is located roughly 30 minutes away by car. And this is a huge barrier for many families in the area who don't have access to a car, and there isn't really reliable public transportation options. And this has led many families to rely on limited food selection available at a local Dollar General and a neighboring gas station, and has contributed to rising nutritional concerns among families in the area. In addition, while there is healthcare available from some wonderful nurse practitioners in the community, the nearest county hospital also requires about a 30-minute car ride for those who are able to afford medical services and who require more specialized care. Additional health concerns in this community include historically high rates of parasite infection in the area, although this hasn't been well studied recently, and environmental toxin exposure, including runoff from agricultural pesticides to many farms in the area. And so both of these can compound existing poor nutrition and negatively impact child development and growth, and with long-term health trajectories. And the COVID-19 pandemic appears to have exacerbated these underlying community health concerns, including, for example, increased demand for feeding programs that are offered by the local nonprofits that we partner with. So the combination of poor nutrition, exposure to environmental toxins and pathogens, including intestinal parasites, and limited access to medical care and relevant health information may all contribute to the development of health disparities in this context. So to look at these relationships, we collected a pilot sample from 24 children aged 6 months to 14 years of age to look for evidence of elevated intestinal inflammation during childhood, and a test of inflammation is related to socioeconomic status and parasite infection patterns. I'll say off the bat that this is obviously a very small sample. This is due in large part to this being a new project, a new field site. And so a significant amount of this time during our first visit was spent meeting with community members and community partners and developing trust. So we do plan to do follow-up data collection as soon as we safely can return to the field. So take these results that I'm about to present with a grain of salt. But that being said, we did see some interesting and consistent patterns emerge during this very preliminary analysis. During data collection, we had parents complete household and child health interviews. They provided information on health history, allergy and autoimmune disease, household makeup, household infrastructure, and factors related to lifestyle and socioeconomic status. Participants also collected and returned first morning stool samples, so similar to what we did in Ecuador. And this sample was used to measure fecal calprotectin, which is a marker of intestinal inflammation that's been linked with inflammatory bowel disease, gastrointestinal cancers, certain bacterial infections, including Helicobacter pylori, and environmental enteric dysfunction. And I performed these analyses using a commercially available enzyme immunoassay kit from Bowman Laboratories. You can see pictured here. And then we also used these stool samples to look for signs of parasite infection, both in the field using microscopy, and then later using 18S rRNA sequencing. And these analyses were performed by our collaborator, Dr. Liz Malott at Northwestern University. So looking at the results quickly, we see comparatively high rates of fecal calprotectin and a high prevalence of clinical elevation in the children in the sample. Even though it's a small sample, we consistently see this pattern. So table 1 here on the right shows the fecal calprotectin levels of all children sampled, and then the sample broken down by age and compared to similarly aged reference samples drawn from the literature. The only sample with higher levels of fecal calprotectin in comparison was a group of children between the ages of 1 and 1.5 from Bangladesh. So you can see pretty interesting that we're seeing such high rates of inflammation compared to the other literature. And this includes compared to the inflammation rates measured in our sample of Shuar, which we analyzed using similar methods. However, during infancy and early childhood, fecal calprotectin levels are known to be variable as the gut develops and diets change. So for this reason, further analyses that I'll present only focus on fecal calprotectin levels in the 20 children who are age 3 and over. Before I move on though, table 2 here on the left shows the breakdown of inflammation levels using clinically established cutoff points. So you can see a large proportion of our sample have moderate to high inflammation levels. And there were no sex differences between fecal calprotectin levels. So looking at indicators of how household income may be associated with intestinal inflammation, we do see some signs that perhaps there is an inverse relationship. And this is from bootstrap linear regression analyses do suggest that household income may be negatively associated with fecal calprotectin levels, although this did not reach significance. But there is an interesting pattern. We need to test this further with larger samples. And then looking at the parasite infection patterns, which we were very interested in when we started this study, as we were analyzing the samples in the field, we didn't see as compared to what we see in Ecuador as clear signs of infection. In part, the sample quality was not as good. There was a lot of degradation, but we did see some signs, for example, of tapeworm infection. So you can see image here in the top right. But the 18S rRNA sequences had some really interesting results. This is all new data. These results were delayed because of the pandemic and lab-related shutdowns. So we just recently got these results, but we did see some clear signs of infection in this small sample. Including infections with Cryptosporidium, platyhelminths like tapeworms, and nematodes, such as hookworm. Interestingly, bootstrap de novo analyses indicate that individuals infected with helminths in general and platyhelminths, like tapeworms specifically, had higher fecal calprotectin levels, higher levels of inflammation than those who were uninfected. So these findings, as I mentioned, are very preliminary and based on a small sample, but we do see consistently elevated inflammation levels here. And I think that the fact that these relationships are being documented during this very early pilot work using a small samples could indicate that intestinal inflammation and parasitic disease may be underappreciated issues among those experiencing poverty and racial equality in the rural southern US. And these findings provide some support for the idea that living conditions associated with low socioeconomic status, such as having lower income levels or pathogen exposure, may contribute to higher rates of intestinal inflammation among children in this sample. We also saw clear signs of parasite infection in this sample, which is very interesting. These results suggest that children living in this area may be exhibiting these really robust immune responses early in life, perhaps due to environmental exposures, including parasite infection, but also perhaps exposure to certain types of bacteria. So the local nurse practitioner told us that there are high levels of pathogenic infections with bacteria, including Helicobacter pylori. So this is something we'd really like to test in the future. And these chronic effects may, we see these patterns coming online so early in life during these key developmental periods. If this is sustained throughout development, this might contribute to the development of prevalent health inequities and poor health later in life, including, for example, higher risk of certain gastrointestinal cancers and other poor health outcomes in adulthood. But further testing is required to clarify these patterns. So to end, I'd like to take a moment to highlight some of the immediate next steps building off the analyses I presented today. Once it is safe to return to the field, as I mentioned, we haven't been able to the past couple of years. So first, in my work with the Shuar Health and Life History Project, when I'm able to go back to Ecuador, as I mentioned, I would like to collect environmental samples to identify areas of the house with more parasite eggs may be concentrated. So for example, taking swabs of the floor, of cooking surfaces, of latrines or outhouses, water samples. And in addition, I would like to collect data from Shuar living in cities, so a much higher level of market integration. And so this could help flesh out the data set and to help us to determine how the higher levels of market integration in these communities may be linked with parasite infection patterns in the population. I also plan to revisit the communities that I highlighted here in this study. I have an engaged community grant to go and revisit the communities and to develop educational outreach materials with local healthcare providers. So as soon as I'm able to do so, I plan to do that. And these visits will also afford me the opportunity to collect longitudinal data. Since market integration has been ongoing, this hasn't stopped in time. There's been ongoing market integration. It'd be really interesting to see how changes over time within these communities may be influencing these patterns that we're seeing with regards to parasite infection risk. Turning now to the REACH study in the United States, when we can go back to the field, we do have funding for another 2 to 3 years of preliminary data collection, and we hope to follow this with additional longitudinal work with partner communities. So we do plan to return to this community in the Mississippi Delta to collect additional data and to do some outreach and present results. But we also hope to visit in collaboration with a regional nonprofit we've been working with. We do hope to visit other study sites in Alabama, in Texas, in Appalachia, and Louisiana. As in Ecuador, we plan to collect environmental samples to better assess where eggs may be concentrated in the community and infect more people. And also, we really would like to start exploring possible routes of zoonotic infection spread. When we were there, we noticed there were a lot of stray domestic animals, cats and dogs in the area. And so it'd be really interesting to look at those parasites that may be impacting humans. We also plan to partner with a colleague to measure toxin and pesticide levels in environmental samples, which I alluded to earlier, but we haven't directly measured. This was a common concern that was identified by participants during interviews, given the farms in close proximity and runoff, and a lot of concerns that children playing outside may be being exposed to these pesticides with implications for health and development and inflammation. And finally, we're really interested in expanding our study scope to examine how cohabitating intestinal parasites and gut microbiota may be interacting to influence intestinal inflammation and immune function with implications for long-term child health and development. So we're currently developing partnerships with microbiologists to develop this aspect of the study. And with that, I would like to end by thanking the many individuals and organizations who made this work possible, especially our participants. And I'm happy to, and I provided my contact information here. So if you have additional comments or questions you don't get to, please feel free to email me. I'd be happy to hear from you. I'm happy to take questions now.
Well, thank you, Dr. Gildner, for a really, really fascinating and enlightening seminar. I'll encourage everybody in the audience once more to please leave your questions in the live chat so that we can relay them to Dr. Gildner. We do have one here already from Rachel Penzikowski, who asks, what is the sewage and sanitation situation?
I think Matt just had some troubles. Really interesting seminar, Dr. Gildner. Thank you again for sharing your research. I will just repeat the question from Matthew, and that was the question from Rachel. So what is the sewage sanitation situation like in the communities where you were working in the Mississippi Delta? I'm thinking of some of the conditions Catherine Coleman Flowers has documented.
Yes, no, thank you for mentioning Dr. Flowers' work. We were very inspired by her studies in Alabama. It's really important work. So we don't, we ask about this and when we collect data, and what we have observed is that there's a lot of reports of flooding and sewage backups. So it does seem to be a very real concern. And this is something we hope to get more into when we have a larger sample where we can start exploring more of these household infrastructure or specific pathways, similar to what I did with the Shuar. We can test these more with our statistical analyses in the future. But we do see some indications in our conversations with participants that sewage backups are, especially when it rains and they're located close to a creek, so there's a lot of flooding that happens, and that can be really harmful if the sanitation systems are not up to snuff, and you start seeing that during flooding, that you're getting some backups. So it's a huge concern. Thanks for the question. It's a really good question.
All right, thank you. And while Matt is connecting again, and we're waiting for more questions in the chat. Oh, here is another question from Rachel again. She says, also, are you or anyone planning to study antibiotic resistance in soil or human microbe samples in these communities?
Yeah, I mean, it's something we're interested in. It's not something we're actively pursuing at the moment, just given the other relationships we're trying to develop. But as part of the microbiome work, that would be very interesting to look at. And we are interested in other types of, evolutionary relationships in terms of allergies and asthma. So some of these types of relationships that might be occurring in these contexts. So yes, it would be really interesting as part of this environmental sampling to look at these types of relationships. But so far we haven't had that set up, but that's a wonderful future direction that would be great to pursue.
Hi again, sorry for dropping out for a minute. I do have a question of my own, which I'm curious about. And you might have alluded to this with your mention of looking at the impact of the presence of domestic dogs and cats on disease prevalence. But I wonder if you know whether your helminth species of interest are specialized on humans or whether they also infect other species, and if the presence, if so, of those species could predict infection intensity.
Right, so the ones we're mostly, for example, the specific helminths I talked about today are mostly focused on humans, but we'd be interested, for example, with the cats and dogs. One of the parasites we'd be primarily interested in would be Toxicara, which is a species, there's one that affects cats, one that infects dogs, but it's known that humans can become incidental hosts. And there's been some interesting studies looking at that. It does seem to be tied, these infection patterns, with low socioeconomic status and environmental exposure. So there's a really interesting study, for example, that was done in New York recently, doing environmental sampling that linked, especially Toxicara cati, the one that impacts cats. And they found the high prevalence of that in parks and outdoors in lower income areas in New York. And they tied that with high stray cat population in those areas. So I think the highest levels, if I remember correctly, were in the Bronx, and the lowest levels were in Manhattan. So that's an urban setting, not a rural setting. So it'd be interesting to see if those are playing out in rural settings too, for that parasite, for example.
Thank you. So thinking in the Shuar community, I was curious, you showed that the children are more prone to have high risk of infection. And I was wondering, what are those effects on those children? And also, if you can compare those effects between children and adults, is going to be the same? The response is going to be different?
Right, that's a great question. We do look at this. I really recommend, if you're interested, checking out work by my colleague, Dr. Sam Erlacher, assistant professor at Baylor. He did some really interesting work looking at energetics in children. So he's got some wonderful papers published looking at the impact of infection, of immune responses in relation to growth. And he's demonstrated there are growth impacts on children. So we are seeing some indications that these infections, especially if you have a heavier parasite burden, may be impacting growth in kids negatively. And then in terms of so the other question was about adults. So for adults, I think there's been, this is somewhere where it'd be interesting to get more at the longitudinal data, where it'd be interesting to see how immune function is changing over time in children and adults. So a lot of the time when we ask about symptoms, these tend to be lighter or more moderate infections that we're seeing. So we're not seeing a lot of those more severe health effects I mentioned, like intestinal blockage or things like that. But it would be interesting to see over time how those infections are playing out in terms of long-term responses. I think that could be interesting to look at in terms of age differences.
Thank you. We have another question here from Barbara Kunkel, who asks, can you tell us more about how the community in the Mississippi Delta feels about your study? What type of reception did you get at first?
Right. So that's part of why I mentioned we did a lot of community building the first time we were there. So there tended to be a lot of, understandably, distrust. I think there's been a lot of examples of, service groups coming through and staying for just a couple of days and then leaving. And so it's, the onus was on us to demonstrate that we came in good faith and we actually wanted to develop partnerships with the community. This was supported in large part because before we went, we were invited to come by the nonprofit that we've been partnering with that does a lot of work in child health and education. So they were really interested in our project and partnered with us and hosted us while we were there and helped to facilitate conversations with community stakeholders. And we're continuing to work with them. So for example, we just this past couple of weeks, we worked collaboratively with this group to co-write a community engagement grant to build off these very preliminary results, to do some development with community members, to develop educational materials, to form a community health council. So these were all ideas that the community partners had. And then to hire some local medical professionals to help us to develop as a group a medical database, since an issue here is if people are concerned by what our results are showing up, we need to establish next steps. And so by partnering with local community provider doctors who are plugged into the communities, have community trust, and also have a better sense than we do as academic researchers of what the actual healthcare options are in the area, especially clinics that work on a sliding scale, then hopefully if we get the funding, we can help pay these paid consultants to help us develop a database that community members can access to get this information going forward. So it's definitely, understandably, a slow process, and the pandemic has really disrupted a lot of this, although we've stayed in contact with our community partners in this nonprofit throughout this time. But in terms of face-to-face interactions, which are so important with building relationships within the community, that's been really curtailed by the pandemic, unfortunately. So hopefully, as soon as it's safe to do so, we can get back and continue developing those relationships.
Great. Here's another comment from Rachel. She's really into your seminar. She says, if you haven't already met Kelly Lane DeGraff at Fondabon, she's studying. Yes. She says she's studying roundworms in soil and parks in St. Louis and in raccoons in those parks.
Thank you, Kelly Lane. All right, I'll look her up. Thank you.
Yes, and with that, I have one more maybe basic question about evolutionary history. If we humans, do we evolve some kind of mechanistic strategy or something to avoid those parasites, like for example, the roundworms or the tapeworms, thinking more in the immune system?
I was going to say too, in terms of evolved responses, my colleague, my co-PI, Dr. Theresa Gildner, just published, or she led a really great paper that was just published in PNAS on disgust sensitivity. And she drew on data and work we did in Ecuador among the Shuar. So that might be one way that we've evolved to kind of avoid these sources of infection. And so it's a really, really recommend if anyone's interested in that. If you look up PNAS disgust sensitivity Shuar, you'll find it. In terms of immune responses, as I mentioned, there is a branch of that immune system that seems to be evolved to deal with these types of extracellular parasites. And so I didn't have time to get into it here, but we do measure immune markers as part of our research. So we've collected dried blood spots. So these are minimally invasive. We do a finger prick, collect a few drops of blood on a filter card, and we can dry them and bring them back to the lab and then analyze them. And so we have looked at different immune markers that way. And so, for example, one of the ones we're very interested in is immunoglobulin E, and that's an antibody that's linked with parasite infection and also with allergies and asthma. So we're interested in for a couple of different reasons. But we do look at relationships, for example, and how IgE and these immune responses linked with parasite load patterns may be playing out in these different contexts. And I didn't present the data here, but this is a recent, again, small analysis based on this small sample, but we do find some signs, some analyses that I've done that the children in this sample in Mississippi had significantly higher IgE, this antibody linked with parasite infection, at higher levels than compared to NHANES data, a similarly aged cohort. So there might be seeing some immune responses that are different in this Mississippi setting, although need to do more data collection and analysis of bigger samples in the future.
Wonderful. Well, we are right at 5 o'clock, so I think we will wrap up here. I'll let everybody in the audience know once more, if you have any questions for Dr. Gildner, please feel free to reach out at the email she's provided for us right now. And thank you, Dr. Gildner, for joining us. This has been a really fascinating seminar and conversation here at the end. And thank you all in the audience for tuning in, and we hope you join us next week.
Thank you very much. It's a pleasure.